Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about post-translational modification which takes place after the translation process during protein synthesis. So it is common for both prokaryotes as well as eukaryotes. So post-translational modifications refers to covalent chemical alteration of proteins after it is being formed or manufactured by ribosomes and its synthesis are complete. Okay, these modifications expand the functional diversity of proteome or all the proteins by altering protein activity, cellular localization, structural stability, interaction networks without rewriting the genetic code. So it simply modify proteins uh, and after the modification the protein can do a different activity, some special activity. Okay, so at first phosphorylation, modification by phosphorylation, that means addition of a phosphate group. Generally phosphate group, the attachment of phosphate group is added by is dependent on the enzyme kinase and phosphatase which help to remove phosphate group. Okay, so it is one type of modification. Next one is glycosylation, attachment of carbohydrate group or carbohydrate chain to nitrogen atom or oxygen atom of a specific amino acid. So do you know the structure of amino acid? It consists of an alpha carbon with carboxyl group, amino group, hydrogen and one side chain. So either it will attach with this nitrogen or to oxygen. This step is critical for proper protein folding and immune system antigen during antigen recognition. Okay, either it will be a N-linked glycosylation or it can be a O-linked glycosylation. Next, amino acid glycosylation can be N-linked, O-linked, or it can be C-linked or S-linked. N-linked glycosylation generally target the amino acid asparagine and attachment site is the amide nitrogen atom on the side chain. Typically occurs at the consensus sequence asparagine X, which can be any amino acid except proline, serine or threonine. It can be O-linked glycosylation. Apart from these two glycosylation, it can be a C-linked glycosylation which target tryptophan amino acid and attachment site a rare modification. It is very rare modification where sugar attaches directly to carbon atom on the tryptophan side chain. S-linked glycosylation is there and target amino acid is cysteine, attachment site the sulfate atom on cysteine side chain. Okay, so these are different types of glycosylation method. Then ubiquitination. Ubiquitination, it is covalent binding of ubiquitin, which is a small regulatory protein, to lysine residue. Polyubiquitin chain target aging or misfolded proteins to the proteasome for degradation. So generally this ubiquitination leads to the degradation of proteins. So at first attachment takes place, a chain of ubiquitin tags or marks the old, damaged or broken proteins. Then targeting the tagged protein with ubiquitin moves towards the cell's waste disposal unit. Then finally degradation, the proteasome breaks down the tagged protein into small pieces. Next, acetylation. So it is the transfer of acetyl group to lysine residue or N terminus of a protein. So histone acetylation alters chromatin architecture to drive gene transcription. There are two major types of acetylation. N-terminal acetylation occurs early during protein translation on protein's front end, help to stabilize the structure. Lysine acetylation, it is the reversible modification on internal lysine residues that changes the charged properties. Okay. Next, so lysine acetylation, it adds acetyl group on the side chain of lysine residue. It removes the positive charge of lysine. It relaxes tight DNA histone ties to turn on gene. It is reversible process controlled by enzyme. Lysine acetylation. And N-terminal acetylation attaches acetyl group to the very start alpha amino group of protein chain, often happens while the protein is being made during translation. Helps control protein lifespan, folding and location. It is generally treated as a permanent change, unlike the lysine acetylation. Next one is methylation. It is the addition of methyl group to arginine or lysine side chains, highly prevalent in epigenetic regulation to activate or suppress or repress a specific gene. So epigenetic regulation means we can change, or the environment can change the activity of a gene without altering its genetic code. That is called as epigenetic change or modification. There are two types of methylation. Lysine methylation, catalyzed by enzyme lysine methyl transferases and reversed by demethylases. It acts as a docking site to recruit effector proteins that activate or repress gene transcription. Arginine methylation, catalyzed by protein arginine methyl transferases. It regulates RNA processing, signal transduction and transcriptional activation. Okay. Next, lipidation. It is post-translational modification where lipid groups attach to protein. This change makes protein more oily, helping them to stick to the cell membrane, move to correct place in the cell and interact with other molecules. So key steps include prenylation, myristoylation, palmitoylation. Okay. So what is prenylation? It is the adding of isoprenoid chain to cysteine residue at the end of the protein. Then myristoylation, it is the attaching of 14 carbon myristic acid to N-terminal glycine. It is often a permanent change. S-palmitoylation, it is the addition of 16 carbon palmitic acid to a cysteine residue, this change can be reversed by the cell to control protein activity. Then GPI anchors, attaching of complex glycolipid to end of protein to tether it to outside surface of cell membrane. So this is the case of lipidation. So now the proteolysis, it is the cleavage of peptide bond. It cuts away inhibitory signal sequence or prodomains to activate proteins like to mature insulin. So how this proteolysis works? Without catalyst, breaking a peptide bond naturally can take hundreds of years. Biological systems use targeted mechanism to make this happen quickly as well as safely. So the enzyme called protease, such as pepsin, trypsin, they target specific protein and cut specific peptide bonds. So it can be limited or complete proteolysis. Limited proteolysis cut a specific site to activate the protein, while complete degradation breaks the protein down into raw amino acids. Then ubiquitin proteasome system. here cells tag unwanted or damaged proteins with molecule ubiquitin. A barrel shaped structure called proteasome then destroys that tagged protein, or it is called as ubiquitin aided proteolysis or protein degradation. Okay, so it is all about your post translational modifications which takes place after the translation process, after the protein is manufactured or formed. Okay, so with this I am concluding my lecture video here. I hope this video will be beneficial for you all, and if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.